Besides drugs and fava beans, what else can trigger this?
A few things. The paper talks about 4 main clinical manifestations.
Clinical manifestations. You mean how it shows up in real life?
Exactly. So first is drug-induced hemolysis, which we already talked about. Primaquine, certain antibiotics like nitrofurantoin, methylene blue.
There's a list of drugs you have to avoid?
Yeah, I basically have to check every new medication. It can be annoying, but it's manageable.
What's the second one?
Infection-induced hemolysis. This one surprised me. Turns out, infections themselves can trigger episodes, even without any drugs.
So if you get sick.
I have to be careful, yeah. A hospital study found that infection was actually the most common trigger for hemolytic episodes in G6PD patients.
Third one?
Favism, the fava bean thing. Not everyone with G6PD actually gets sick from fava beans, though.
That's weird. Why not everyone?
That's a good question, and no one knows for sure. The paper says G6PD deficiency is necessary, but not sufficient for favism. There's probably another genetic factor involved.
And the fourth?
This one is actually the most serious. Neonatal jaundice.
Jaundice in newborns?
Yeah, and here's what's surprising. It's not mainly from hemolysis. The bigger problem is that G6PD deficient livers in newborns have trouble processing bilirubin. If it gets severe, it can cause brain damage called kernicterus.
That's scary. Can they test babies for this?
That's actually one of the most open questions in the field. The paper asks whether all newborns should be screened. Right now, it varies by country.
Okay, so this is a part I really want to understand. G6PD is on the X chromosome, right?
Right, it's X-linked.
So explain to me. If guys have one X and one Y, and girls have two X's, how does that work?
Okay, so for guys like me, it's pretty straightforward. I have one X and one Y. If that one X has a mutation in the G6PD gene, I'm deficient. No backup copy.
And for me?
Well, you have 2 X chromosomes. You got one from your dad and one from your mom. One of yours has a mutation and one is normal.
So I'm a carrier, but does that mean I'm half affected?
Yeah, so here's where it gets really interesting. This is actually one of the biggest discoveries to come out of G6PD research. Have you heard of X inactivation?
I think so. Something about one X being turned off.
Exactly. So in the 1960s, Dr. Beutler, who is the author of this paper, was studying G6PD inheritance, and he noticed something weird. Mothers of affected sons who had to be carriers showed really variable symptoms. Some seemed normal, some were intermediate, and some were fully affected.
That's strange. Shouldn't they all be the same?
You'd think so, but around the same time, a scientist noticed that in female cells, one chromosome looked different. It was condensed into something called a bar body.
A bar body?
Yeah, and Beutler had this brilliant idea. What if only one X chromosome is active in each cell? And what if the choice of which one gets turned off is random and happens early in development?
So like some of my cells have the normal X active and some of the mutant X active?
Exactly. You're a mosaic. Some of your red blood cells have normal G6PD and some are deficient.
Oh wow, so that's why carriers have different symptoms.
Right. It depends on the ratio. If by chance more of your cells have the normal X active, you'll see mostly normal. If more have the mutant X active, you might have symptoms.
That's actually really cool, but also kind of random.
It is. And this discovery the X inactivation hypothesis turned out to be huge for all of biology, not just G6PD. It's used to study cancer, understand genetic diseases. It was a really big deal.
Okay, but here's my big question. If I have kids someday, what are the chances I pass this on?
Good question. So if you have a son, there's a 50% chance he'll get your X with the mutation, and he'd be fully affected like me. 50% chance he gets your normal X, and he's fine.
And if I have a daughter?
50% chance she gets your mutant X and becomes a carrier like you. And 50% chance she gets your normal X.
What about the dad? Does it matter if he has G6PD deficiency?
Yes. If the dad is affected, then every daughter will get his mutant X. If you're a carrier and he's affected, there's a chance you could have a daughter who's fully affected with both X chromosomes carrying the mutation.
That's a lot to think about. I guess I should make sure any future partner gets tested.
Yeah, it might be worth knowing. Especially since G6PD deficiency is so common in certain populations. Alright, so this is one of the coolest parts of the paper. So G6PD deficiency is really common in sub-Saharan Africa, the Mediterranean, the Middle East, Southeast Asia.
Those are all places where malaria is common.
Exactly. Scientists noticed this pattern and hypothesized that G6PD deficiency might protect against malaria.
Wait, so having a genetic defect is actually protective?
For malaria, yes. There was this elegant study by Lozado. Remember how female carriers have a mix of normal and deficient cells?
Yeah, the mosaic thing.
Yeah. So he looked at carrier women who had malaria and he asked, which cells do the parasites actually infect?
Ooh, that's clever.
Right? And he found that the malaria parasites preferentially infected the normal cells. The G6PD deficient cells were relatively spared.
So the parasites don't like the deficient cells.
It seems that way. Maybe it's because the cells can't handle the oxidative stress the parasites creates, so they die before the parasites can reproduce. The exact mechanism, though, isn't really totally clear.
So natural selection kept this mutation around because it saved people from malaria.
Exactly. It's called a balanced polymorphism. The mutation has costs, like what I deal with. But in malaria endemic areas, the benefits outweigh the costs.
Evolution is wild. So, how much do scientists understand about G6PD now?
Well, a lot more than in the 1950s. They've cloned the gene, they've figured out its structure, and identified over 370 different variants.
370? That many mutations?
Yeah, and the WHO classifies them by severity. Class 1 is the most severe, people have chronic anemia all the time. Class 2 is severe, but intermittent. Class 3 is moderate. That's what most African American patients have. Class 4 and 5 are mild or even increased activity.
Do they know which variant I have or you?
You can get genetic testing to find out. Different variants can mean different severity and different triggers to watch out for.
That might be useful to know.
For sure, especially with newer drugs coming out all the time. It's important to know your specific risk level.
So, what questions are scientists still trying to answer?
A few things. One is developing better ways to test if a new drug will cause hemolysis. Right now, you kind of have to, wait and see, which isn't great.
That sounds stressful.
It can be. Another question is about newborn screening. That is, should we test all babies? It could prevent kernicterus, but there are costs and logistics to consider.
And figuring out why only some people get fauvism?
Yeah, that's still a mystery. There's probably another gene involved, but nobody's found it yet.
So even after 70 years, there's still stuff to discover.
That's science. Dr. Beutler wrote something really nice at the end of the paper. He said, "It's a rare privilege for a scientist to follow a field for 50 years, from the initial clinical problems through understanding biochemistry, genetics, and molecular structure."
He sounds like he really loved his work.
I think he did. And G6PD research has contributed to so many other fields, understanding X inactivations, studying cancer, population genetics, etc.
Pretty cool that something affecting you personally has such a big impact on science.
Yeah, I think about that sometimes. It's not just a condition I have to manage, but it's connected to this whole fascinating scientific history. If you're a patient like me, you need to watch out for certain drugs, foods, and infections. If you're a carrier like Anna, you might have some mild symptoms and could pass it to your kids.
And the reason it's so common is because it actually protects against malaria, which is kind of amazing when you think about it.
If you want to learn more, check out our website, g6pdresearch.org. We've got a database of all the variants, a world map of prevalence, and links to research papers.
And the original paper we discussed today is linked in the description. It's by Ernest Beutler, published in Blood Journal in 2008.
Thank you so much for watching. If you learned something, hit that like button and subscribe. We're going to make more videos about G6PD and other science topics.
And if you have questions, drop them in the comments. We'll try to answer them.
See you next time.
Bye, guys.
We're finally done.